So now we've seen how countercurrent multiplication can lead to an osmotic gradient in the interstitial fluids in the medulla, and that means that we can now take a look at the fate of urea. In this video, we're also going to cover the fates of other solutes and how blood flow factors in. But before we get into any of that, I want to note that out of this 1200 milliosmol gradient down here, only about 600 milliosmoles of this gradient really deep in the medulla is actually due to sodium. So the other half of that is actually due to urea. And so we need to talk about how exactly that works. So, as with the amphibian kidney, urea gets added to the nephron at the glomerulus through ultrafiltration. And remember in the amphibian kidney that there is some degree of concentrating of urea in the amphibian nephron simply by virtue of the dynamics of the proximal and distal convoluted tubules being permeable to water and then there being active transport of salts back out of the tubule, right? So again, so in the amphibian nephron, urea gets concentrated because salts get actively transported out and then water follows and urea can't, so therefore the concentration of urea winds up increasing. So that's the amphibian kidney. But things are a little different for mammals. Instead, there are certain specific locations along the nephron in a mammalian kidney that are permeable to urea, and that facilitates further concentrating of urea. So where are those locations? So those specific locations are here along the collecting duct. So we've got urea channels here. But these urea channels are only found along the inner medulla, right? Okay. And so what's the consequence of that? So urea is going to get concentrated by processes that remove water from the urine in the loop of Henle, right here. And that's going to favor movement back out of the collecting duct, right? So if the urea is more concentrated, by the time the urine reaches the collecting duct, then that's when there are urea channels present, that's going to favor the movement of urea out of the collecting duct. And so then that's going to, in turn, favor an increased concentration of urea in the medulla. And that's especially going to be the case under conditions of antidiuresis, right? A little hard to see the red here, or blue here, but so there's going to be aquaporins as well, right? So water is going to be able to move. So those aquaporin proteins are going to allow the movement of water back out, and that's also going to favor additional movement of urea. But so far, that doesn't really seem very helpful for getting rid of urea, right? And there's going to be a limit to this diffusion process. So here's the real kicker to this whole system. There are also urea transport proteins in part of the ascending loop of Henle that we haven't talked about yet. So they're found in the thin ascending portion. So you've got more urea channel proteins in here. Okay? And so what that does is that means that the urea that makes it here into the medulla can diffuse right back into the urine. Right? And that's diffusion. But you might still be wondering, okay, now we've got more urea over here also, but how's that really going to help? Why might that help? And the best analogy for thinking about this is to think about if we're imagining traffic traveling on a highway, but it's going to be a highway where there is a loop added into that highway. Okay. Okay, so if we've got a bunch of vehicles traveling along here, and some of them wind up on this loop over here. Some are going to keep going, but some are going to wind up on this loop, and they might loop around once, they might loop around twice, they might loop around a couple of times. So what's going to happen if we introduce this loop is there's going to be a buildup on this loop. And so the equivalent thing is going to happen here with urea in the loop that's been established right here, right? So yes, some urea is going to travel around, but the net effect is going to be a buildup of urea at this point in the kidney. Okay, so this isn't really good for transportation design, but in this case it's really useful because this buildup of urea here in the inner medulla means there's going to be this larger interstitial concentration gradient, right? In people, so that's builds us up to 600 milliosmoles. And that's going to favor the retention of water, and it's going to facilitate further concentration of the urea that departs in the urine. So boom, how amazing is that? So I now want to note that the net consequences of this buildup of urea in the interstitial fluid mostly just facilitates urea excretion. This part does not contribute directly to water reabsorption. And that's because when there are channel proteins, as here and here, that allow free movement of a solute, that solute is no longer going to be osmotically active, right? So even though we might have an osmolarity overall down here that's 1200, this fraction of that osmolarity is not osmotically active. So this urea buildup here is not contributing to the movement of water. And instead, it's really that active transport of sodium that happens here in the ascending limb of the loop of Henle that's the mechanism behind water concentration. I hope you've got that. If you're still struggling with it, it's worth going over this a few more times until that starts to become more clear to you. And it can take time because it's a very challenging concept. Okay, so now we're going to briefly take a look at dynamics for other solutes. And for this, I'm going to focus just on glucose, potassium, and chloride just a tiny bit for the moment. But as with the amphibian kidney, realize that there's active transport of amino acids back out of the proximal convoluted tubules. And also be aware that divalent ions, such as calcium and magnesium, are also going to get harvested back out of the urine. And again, we aren't going to go into cellular details for those divalent ions, but if that winds up being an area of interest for you, there's plenty to learn in that arena. Okay, so for potassium and glucose, what we're going to do is a close-up examination of the transport protein configurations that facilitate reharvest in 3 spots. So those 3 locations are in the proximal convoluted tubule, then in the thick ascending limb of the loop of Henle, and then in the collecting duct. Okay, so to start out with, let's take a look at a cell that's up here in the proximal convoluted tubule. Here we go. Okay, so we're going to walk through these transport proteins one at a time. So first over here on the basolateral membrane, we're going to start with our friend, the sodium-potassium pump, and you'll notice there's also a potassium channel over here again. And you'll also note that in this case, there are potassium channel proteins depicted on both membranes. However, in this tissue, think of this as just permitting free movement of potassium, but without major consequences for potassium balances here. So no net potassium movement in the proximal convoluted tubule, okay? And so what that means is our major focus here at the sodium-potassium pump is going to be what we saw previously, that this is going to establish a sodium concentration gradient. And, with sodium higher here in the interstitial fluid and sodium concentrations lower here, and favoring a movement net movement of sodium into the cell, and that's going to facilitate glucose reabsorption, again using a secondary active transport mechanism. So illustrated right here, right? Co-transport of sodium and glucose back in, moving sodium down its concentration gradient. And it's actually the sodium concentration gradient that also facilitates the reuptake of amino acids, and that's not shown in this picture. This figure also highlights how the proximal convoluted tubule can be involved in regulating blood acid-base balance as well, right? So here's an excretion of hydrogen atoms, and we've got a bicarbonate cotransporter also. But again, we're not going to dive into that topic in depth, but be aware that the kidney is involved in regulating blood acid-base balance. Okay, so this is the proximal convoluted tubule. So now in comparison, here's the transport protein arrangement that's found, again, in the thick ascending limb of the loop of Henle. So here you can see, as I promised, that there's this NKCC co-transporter, and it's again powered by a sodium-potassium pump. And in this case, there will be more net movement of potassium back out of the urine, along with a corresponding reabsorption of chloride. So that's here. So I'm going to put here the net potassium direction of movement is going to be back into the interstitial space. Okay, so that's at the thick ascending loop of Henle. So lastly, here is one of the cell types that's found in the collecting duct. This is a sodium reabsorbing cell, or it's also called a principal cell. And the main thing that I want to note here is just that the collecting duct, along with the distal convoluted tubule, is a major point for control and regulation of potassium ion balances. So we're going to put potassium ion balance regulation point. Right, so this is where there is control over how much potassium gets added to the tubular fluid, partly through these passive transport mechanisms and partly through active transport. It's not illustrated especially well with this particular cell, but again, those are the major points for the collecting duct and for thinking about potassium retention or excretion. Okay, so that's it for the other solutes, which means that the final topic I'm going to touch on for this video is going to be blood flow. And here's what I want you to think about. Okay, so all these cells that are deep in the kidney here need to get oxygen, and they need to have mechanisms to get energy and get rid of waste products, just like every cell anywhere in the body. So they need blood flow. But on the other hand, blood vessels tend to be fairly leaky by nature. And even blood vessels that aren't fenestrated are structured to facilitate the movement of solutes and gases. So if we run blood vessels deep into the medulla all willy-nilly, we risk undoing all of this hard work done by the loop of Henle and the collecting duct to establish these concentration and osmotic gradients for salt and urea. So that's a big problem. And so that's why I have these capillaries over here on the left of this figure illustrated in this fashion, because that reflects how blood vessels are oriented within the kidney. They have this really specific structural arrangement to reduce those potential issues of leakiness. So these blood vessels are long and they run in parallel to the loops of Henle. And they're referred to as the vasa recta, which would be Latin for straight vessel because of this orientation, right? Okay, and the benefit here is that so long as blood flow is relatively slow, and it is relatively slow, and it's relatively slow in part because, as you see here, we've already traveled through a capillary bed in the glomerulus, so blood flow is going to be really slow through here. What that means is fluid in these vessels can undergo countercurrent exchange with the interstitial fluid as the blood in these vessels travels down towards the inner medulla and then back up and out again. So here we actually have yet another example of countercurrent exchange. Right? So the osmolarity is going to be really high inside this vessel down here, but then as the blood travels back up and it hits interstitial fluid, that's only at 300 milliosmoles as it travels back up, it's going to freely exchange solutes and liquid, and it's going to return to a lower osmolarity of 300 milliosmoles as it comes out and it leaves the kidney. And so altogether, this long and loopy path ensures that the blood flow doesn't simply counteract all of this other kidney action. Okay, so that's how the mammalian kidneys work and manage to produce those incredible osmotic U to P ratios that I described previously. We're getting pretty close to the finish line now and ready to shift over to talking about excretion in insects and about the diversity and how different types of animals deal with and excrete nitrogenous wastes.